about some protein misfolding. Instead of having cases where the protein folds up correctly and does what we like, cases of protein misfolding. Now, some of these you've probably heard of. Alzheimer's disease affects many families. My wife's mother has it. My own mother had it. Parkinson's disease you may have heard of. And so the two that we're going to be talking about are Alzheimer's disease and this fourth one, transmissible spongiform encephalopathies. An example being the mad cow disease. The protein that's misfolding in the case of Alzheimer's disease is a protein which forms deposits of beta sheets, precipitates in our brain. So this beta here is related to that beta sheet structure. This one here, the mad cow disease, is a prion disease. It's created by misfolded proteins causing other proteins to misfold. And those are the two we're going to talk about. So let's begin with Alzheimer's disease. Both of these involve beta sheets. And folding that creates beta sheets that we don't want. This is an electron microscopy slide of brain tissue from a human with Alzheimer's disease. And you can see these plaques, these precipitated aggregates, these solid precipitates in the brain tissue. It's caused by deposits of misfolded protein fragments, which have aggregated together to make a beta sheet. These are called plaques. You know, like you get plaques on your teeth, you have these deposits. Now there is in the outer membrane of our nerve cells a protein which crosses the membrane. It has a segment on the outside and a segment on the inside, and then a nonpolar segment that goes through the lipid bilayer. And this is called the amyloid protein precursor, the amyloid deposits, the precursor to that. We have enzymes in our cells that sometimes will cut this protein into pieces. And if it cuts off if it cuts off these two sides, then it can produce this nonpolar, this small peptide, this fragment, which is the nonpolar part that just sticks in the membrane bilayer. And those that will aggregate with each other come together and make, nonpolar objects aggregating, make this beta sheet, which is called this amyloid deposit. So they aggregate to form these insoluble fibers of these sheets, and that's what then getting deposited in the brain and is believed to contribute to Alzheimer's disease. These deposits are observed in patients with the disease. A possible drug treatment might be to try to find an inhibitor of the enzyme. If you could find something that would block the enzyme that produces these fragments and prevent the fragments from being made, that might help. Now, so far, people haven't succeeded in doing that. There have been some drugs that have been tested and were in clinical trials and made it through stage 2 of clinical trials, but then they found that there were bad side effects, and they had to stop the trial. It was clear the drug was going to cause more problems than help. And so people are exploring this idea as a possible treatment. So that's just one example of a misfolded protein involving a beta sheet and approaches people are thinking about for treating it. Then mad cow disease is caused by what's called a prion or prion. And what was interesting about this was when people were first discovering it, they couldn't believe what they were seeing. They were finding that there was what looked to be like an infection, something that could spread or become a bigger and bigger issue, but there was no nucleic acid involved. So, we think of infections like this cold I have, that there's some virus and it has DNA or RNA genetic material which can give reproduction of the virus particles, make more and more particles, and replicate and cause the infection, whether it's bacteria or virus doing that. But we don't normally think of, we hadn't before this, thought of a protein somehow causing an expansion of misfolded proteins. So this particular protein in the mad cow disease, called a prion, the word prion stands for a protein-like or proteinaceous infection. Instead of nucleic acid-caused infection, a protein-caused infection, if you want to think of it that way. And it is a very nonpolar sequence. It normally folds into alpha helices and has a long unfolded tail on it. And that form is not harmful on its own, but it is able to refold in an entirely different way, making a beta sheet structure. And once it does that, here we have this alpha helix form, a non-infectious prion protein, PRP for prion protein. If it rearranges, we don't know exactly how this happens, but sometimes these proteins may be caused to adopt a beta sheet conformation. And then those are called infectious. Now they don't cause new proteins to be made, but what they can do is collide with the alpha helix proteins and cause them to also misfold. So, sort of passing on your bad features to a neighboring protein. So one protein might bump into an alpha helix version of the same sequence and then cause it to become the beta sheet. Now you have two beta sheet molecules, and they'll float around in solution and bump into healthy proteins that are alpha helices and cause them to become sheets. And you have two good ones, two bad ones make two good ones, now into bad ones, now you got four bad ones. And just sort of keep multiplying like that. They convert proteins with functional alpha helix conformations into these beta sheets that are bad. The beta sheets here are these blue regions. Now there's a little cartoon, just a little cartoon here I'll play for you, which shows this. I did have it. Here we go. So this green, this is using traffic light colors. Green is good for go, red is bad for stop, and the red protein version will have a sheet, and it's going to run into the green one and cause something bad to happen. We'll just get started here. So there's a healthy one snuggling up next door to this sheet one, and it's causing it to make a sheet. is just a cartoon. And so now it's going to then shade the second one red once it looks like the first one. It's refolded into a bad conformation. Now you have two bad ones, two red ones. And they're going to go out into the solution and each pair up with a healthy alpha helix version, four collide with four other good ones and make them bad. Now you've got 8, and on it goes until you develop these misfolded versions of that protein, which then make deposits in the brain of the cow. It actually destroys the cells and causes then holes or gaps to occur in the brain, and the brain then sort of takes on the characteristics of a sponge, some object with holes in it. So if I can get to my last slide, that's where this name spongiform comes from. And it's transmissible. It's a protein that does it. And the last word just is sort of a neurological disease, encephalopathy. Now the way this was happening was, or it was being spread, was that after cows had been slaughtered to get the beef to be sold to humans, there would be leftover parts of the cow, including their brains. And these would be ground up and used as cattle feed. Instead of just throwing away the materials, there's protein, in the brain. Just grind that up and then use it as cattle feed for other cows. And it was that aspect of feeding the brain tissue of a diseased cow to a healthy cow that would cause this disease to spread within the farms. It wasn't too long ago that this was happening in Great Britain. And then they had to slaughter or burn and kill off a large amount of the cattle that were in Britain to try to save the other ones that were still there until it was understood. So this is another story. There are human-like diseases that are related to mad cow disease. It's an interesting phenomenon, this protein folding and misfolding. Now we're going to start then. I'm going to try to go, I know I won't make it through everything today, but it turns out that Friday's lecture is a little shorter. So we'll keep going. I'll pick up on Friday what we don't do today, and then try to complete our discussions of hemoglobin